Well, hello everyone, and thank you so much for the invitation to be here. Any opportunity I get to talk about microorganisms, I try to jump on that opportunity because I'm pretty passionate about them. And that passion actually started when I was in high school. So I'm kind of fortunate in that I was one of those individuals that had a really awesome biology teacher in high school, right? Everybody I talked to, it's like it was a great experience taking biology in high school, or it sucked. Okay, so I was on the awesome side of things. And in this biology class, I remember my professor had a section focusing on what role microorganisms play in sewage treatment. And back in the day, we called it sewage. We didn't call it wastewater or domestic wastewater like we do now. And I was fascinated by this idea that microorganisms can be used to basically take poopy water and clean it up to the point that it could be returned and used as drinking water. And that's where the passion began. Now, I've been at CU Denver for about 20 years, and then certainly my graduate career leading up to that. We can add a few more years onto that. And I came about within the field of microbial ecology, so I'm a microbial ecologist. And I started working in this area of microbial ecology when microbial ecology was not a very well-respected discipline within the field of microbiology. As I started off with a graduate student, and even in my early years here at CU Denver as a microbial ecologist, I've had numerous experiences where some of my medical microbiology colleagues really sort of poo-pooed the work that I did. And part of that came from this idea that in the field of microbiology, there was this perception, and at the time the tools that we had available largely supported this, that you had a microorganism and it caused X. And a lot of our focus at the time was on disease or medical microbiology. So there was this cause and effect relationship with microorganisms. Now, as a microbial ecologist, I'm an individual that really looks at how microorganisms interact with their environment. So truly looking at the ecology of different types of microorganisms, where they live, why they live there, what influence do they have on their environment, and likewise, what influence does the environment have on them? So contextually, I'm very rarely ever focused on one microbe. And that's been traditional in my training. I'm always thinking in the larger context of how microorganisms interact with each other as they interact with their environment, and their environment interacts with them. And I've been called all sorts of names at professional microbiology conferences because a lot of what we look at in microbial ecology can't be clearly defined. It can't be enumerated. We can't quantify it. And the predictions that we have of the relationship, our relationship with microorganisms, the relationship that they have with us, both directly as humans, but then also in our environment, can't always be clearly delineated. And they're fluctuating on a constant basis. So that provides some context. Now, as a microbial ecologist right now, one of the things that I absolutely love about being a microbial ecologist is I'm not stuck in a box. I can study environments and the microbial relationships with them all across the board. And so while I started off with this passion for wastewater treatment, that developed into basically a passion for how microorganisms can degrade chemicals in the environment in general. And that started off with looking at abandoned mines and acid mine drainage. Actually disturbingly beautiful sites to be at. If anybody has an opportunity to look at an abandoned mine, take a look. It's depressing on the one hand, but on the other hand, the microbiology that's going on is just truly amazing. And then from there, that expanded into other types of environments, asking the question, what do microbes do with these chemicals that come into their environment, and how can the microbes then influence the chemicals once the chemicals are in the environment? So projects that I have underway that kind of deal with this particular topic include not only acid mine drainage, but we also look at waste coming from other types of industry. We look at some of the agricultural industry and the insecticides and pesticides that are released, and how microbes can help us address the toxicity that's associated with the release of these chemicals. We have other projects where we're actually looking at exposure to toxicants on the skin. So we have a pediatric dermatology study where we're looking at early exposure of these children to different types of chemicals that are altering their skin flora, and basically tracking them through adulthood to ask the question, is this early exposure to chemicals altering their skin flora are going to ultimately lead to longer-term disease or pathology. We look at microorganisms that can use some chemicals to degrade waste to see, can we couple them with electricity production? So some of you might have heard some of the recent stories that have been coming out of the University of Buffalo, where they're using microbial fuel cells. And they're using domestic wastewater as an inoculum to isolate bacteria that can produce electricity. And the idea is, can that technology be expanded so it becomes a sustainable technology to reduce our reliance on fossil fuels, but also can you couple waste treatment with it and detoxify chemicals? So I get to study a whole suite of different projects. And so what I want to share with you today, though, is some of this unintended impact that we're having as we're increasingly releasing a number of chemicals into our environment. So while we are not going to focus heavily on the microbial response to those chemicals, as a microbial ecologist, what's coming is forcing us to ask the question, what impact are these chemicals having on microorganisms in general, on the more global scale, and what sort of global or larger societal or health impacts are we expected to potentially see from that? So let's talk about what microorganisms are in the first place. And this is just a really general slide here. First of all, they're the most beautiful things you've ever seen. When you look at them underneath a microscope, the morphology, the structure of these organisms is really incredible. And we all have an intimate relationship with every single group of microorganisms that exist. And I'll talk a little bit about those. So by definition, microbiology is the study of organisms that are too small to be seen with the naked eye. So the human eye has the ability to resolve down to about 0.1 millimeters. And anything less than that, we have difficulty actually resolving detail that's smaller than that. So for the most part, when we're looking at the single cells of the different microbial groups, we can't see them. And we have to rely on a microscope to assist us with that visualization. And all of these but one image is taken with a microscope. So these are micrographs. So fungi. This is one of the major groups of microorganisms. And we all know and love fungi because we like to get mushrooms on our pepperoni pizza, right? And a lot of people don't realize they're eating a fungus when they're eating mushrooms. Now, fungi, other representatives, include the little fuzzy stuff that's growing on your leftovers in the refrigerator right now. That's a fungus called molds. Both are very prolific in the environment. Some of you might be going out for a beer after the talk tonight. So yeasts are another example of the fungi. And the interesting thing is that all members of the fungi participate heavily in organic degradation. And they're some of our primary degraders of organic chemicals that we're releasing into the environment, as well as their role in the degradation of plant material and animal material once it starts, once death has occurred and the material starts degrading. So it turns out they're hugely, hugely important to us. And mathematicians have figured out that if we didn't have the fungal group, if we maintained somehow all the other microbial groups, we got rid of all the fungi globally, we wouldn't be here. Because the amount of organic material would have built up to the point that it wouldn't really be able to sustain life. It's certainly not life in its current form. So we have the fungi. Now, those of you over here on the side might have a hard time seeing this. We also have the algae. And this is probably the most understudied group of all of the microorganisms. Whoa. And nobody likes to really study the algae by and large. We like to study their impact or maybe like what their presence might mean. Some of you might know that when you have a eutrophied system, a system that's got a lot of nutrients coming into it, you can have an algal bloom. Cherry Creek Reservoir, for example, experiences a minimum of one, but usually 2 or 3 algal blooms every summer, to the point where they actually have to close some of the swimming beaches. And it's just due to a lot of organic and nitrogen input into that system. And the algae, though, are participating in photosynthesis. So they're critical to our survival because they produce oxygen just like the plants do. As a matter of fact, the algae actually produce more oxygen than all of the terrestrial plants put together. So they're actually a more critical component to our lifestyle as aerobic or oxygen-breathing organisms than even the terrestrial and the aquatic plants put together. And nobody studies the algae really because they don't cause disease. They don't infect us. They don't cause deadly diseases in humans. There are a few that could produce neurotoxins, and certainly that has an impact, but that's not an infectious disease. So we kind of ignore the algae. And yet the interesting thing is that the algae are highly susceptible to toxic chemicals. So as we start talking about these microbial groups, I want you to think about, what is this impact that some of these toxic chemicals, toxic to the point where they might start negatively impacting the microbial groups? What is that going to look like if we continue to sustain this increased input of toxic chemicals into the environment and we start losing some of our microbial colleagues? So we have the algae. Now, this one is cool, this little fuzzy guy here. This is actually a Paramecium. So this belongs to another group of microorganisms called the Protozoans, or the Protozoa. And the Protozoa are incredibly diverse. Their function is organic carbon degradation. So they like to degrade complex carbon, such as cellulose is a good example. But they can also do other things, like they can ingest cells. This one in particular, this little hole right here, is called a gullet. And what this organism does is it swims around basically with its mouth hanging open, and it consumes other microorganisms that are in its environment as it's swimming around. The organisms get trapped in the gullet, and once they're inside the cell, the organism can start digesting those other organisms. So a critical role, though, in organic degradation, as a matter of fact, the protozoan are the most representative microbial group in the guts of termites, for example. We know that termites feed almost exclusively on cellulose, and while we wouldn't want them to degrade our homes, that's bad, out in nature they serve a critical role in, again, degradation of organic material, think dead plant material. The protozoa, by and large, are also another susceptible group to toxic chemicals, to some groups of toxic chemicals that get released. Over here is an example of a virus. Viruses have a lot of different morphologies, and this certainly is one of the groups that a lot of us get really worried about, and that's actually going to provide a foundation for today's discussion. So the viruses, right? You know, cold, when you get the cold, that's caused by rhinovirus. If you get the flu, that's the influenza virus. There's a long list of if we just focus on the human illnesses that are caused by viruses. And we as humans have a long, fearful, dreadful history with our interaction with viruses by and large. And it's really in part set us up for where we're at today and our view of microorganisms today. Now, many of you might be thinking, what role do viruses play in the environment, right? I mean, they're obviously out there to get us, but they're not. Because it turns out that easily the majority of viruses that we've discovered so far, and there's a lot we don't know about them as a group, don't harm humans, and don't even harm animals, and don't harm plants. The majority of viruses that we've discovered so far are actually viruses against other microorganisms. Okay? Now, this slide up here are the bacteria. And in particular, the bacteria belong to a group of microorganisms called the prokaryotes. And there's now two groups to the prokaryotes. There's the bacteria, and then we have another group called the Archaea. Now, when I was a microbiology student, way back when, we won't reveal the dates, we didn't learn about the Archaea. So we said prokaryotes were all bacteria were prokaryotes. Now we have a second microbial group in there called the Archaea. And being very simplistic about it, the Archaea are considered our extremophile bacteria. These are the organisms we would isolate, for example, from a hot spring or a hydrothermal vent. So they're living under these conditions. We initially thought, when we defined this group of microorganisms, that they lived in these really extreme environments, temperature, pH, radiation. Now we're actually finding them everywhere. I mean, they're going to be all over the place in this room, and this room would not be considered an extreme environment, at least by our definition. So we're still learning a lot about this particular group of organisms. The bacteria, though, everybody in this room knows a bacterium, because we're always hearing on the 9, 6 o'clock, 10 o'clock, whatever it is, news. Maybe people don't watch the news anymore. I'm older. When I say that, my students are like, the news comes on TV? I'm like, oh yeah, I forgot. Okay, everybody pull out the cell phone and, open your Facebook and get the news from Facebook. But on all these newscasts, we're always hearing about foodborne outbreaks. And the majority of those foodborne outbreaks are going to be bacterial related. So E. coli, Salmonella, Campylobacter, and we have regular foodborne outbreaks that we're hearing about in the news. Okay. This is why you all cook your chicken. Don't leave the potato salad sitting out on the countertop for too long, right? You're a little suspicious if you go to a buffet and there's like little brown crusties that look like they're dry on the food. It's this group of organisms that we have an experience with that has taught us that level of concern. Okay, so these are all of the groups of microorganisms. Okay? Microorganisms are ubiquitous in the environment. They're everywhere. And I like this picture because this is a child's handprint, and we could easily just substitute each of our own hands. And it was pressed onto, this is a Petri plate, this round thing is a Petri plate, and in here, it's hard to tell because it's colorless, is nutrient agar. Okay, it's a growth medium that we use to support the growth of different types of bacteria. And all of these different colors and different shapes that we see are colonies of primarily bacteria that are growing. And this is just off of the human skin. This was not like a hand that hadn't been washed in a really long time. We could all create a very similar looking handprint. And so when we look, this is just another image of bacteria in particular, because that's a lot of where our discussion is going to be focused on today. And here's another image of us growing them in the laboratory. They come in all sorts of beautiful colors. They come in a couple different shapes or morphologies that are associated with them. Here, these are round. These purple ones are round. They're called cocci. These red ones here are sort of rod-shaped. My son likes to call them hot dogs. He spends a lot of time with me in the lab. And those are just two different shapes of bacteria. Now, one of the things I want you to note, though, is even if you were a microbiologist, and some of you may be, you can't look at this image and tell me the species of the bacteria that are here. So one of the major limitations that we have in the field of microbiology that, again, is going to contribute to this later discussion that we're going to have is even though we need the microscope and we can use the microscope to see these different microorganisms, when we see these different microorganisms underneath the microscope, that does not give us enough information about them to be able to identify them. The only way right now, at least today, that we have available to us to try to identify the species, which is critical, right? Because you want to know, or your doctor wants to know, did you just get infected with Salmonella or E. coli, or Escherichia coli? That gives you species level information. That's important. Because if you weren't infected with those organisms, your physician's going to go in a completely different direction as to the cause of your gastrointestinal upset, for example. So the way that we have to try to, the tool that we currently use to identify these organisms is we basically isolate their DNA. And then we have to sequence portions of their DNA. And that can give us identity. But that process requires technical training relative to a microscope or to even trying to grow these organisms, which, by the way, even though these two colonies might look the same, they're the same color, they're about the same size, they might have the same consistency if we were looking at them live, we still can't get species level information here. Okay, so just because you see an orange colony doesn't tell you definitively who that organism is. So we have to rely on this genetic sequencing. It's expensive. It's not widely used by the majority of industries that we have that are currently trying to monitor for microbial loading in products. The food industry, for example, I worked for Campbell Soups for a while as a student because I was exploring different areas to study, and I worked, does anybody here work for Campbell Soups? Okay. All of our food, this really isn't unique to Campbell Soups, but all of our food is loaded with microbes. And that's not necessarily a bad thing. Okay? It's actually a testament to how flexible these organisms are with the different environments they're going to survive in. So the food industry doesn't use genetics to try to identify potential contaminating organisms unless they've been linked with a foodborne outbreak. And usually CDC is getting involved, Center for Disease Control. They're getting involved at that point. They're the ones that are really using the genetic techniques to try to figure out, everybody seems to be getting sick because they ate salad at such and such a buffet. We got to try to figure out what the causative organism is. Even in the clinical lab, when you donate a sample and you put it on the little spinny and it goes to some mysterious back room somewhere, right? If you've donated a sample that they might be looking at for a microbial load, the methods that they're going to use initially include the microscope to try to do an early visual characterization. They might try to grow the organisms on different types of food sources to try to narrow down the options. And that's all they're doing is narrowing down the options. Then it's a best guess by your physician. Just because of the limitations of the methods that are being used in that scenario, it really is a best guess. And in many cases, that guess is a solid, it's a good one, right? Because we're going to repeatedly start, as humans, present with similar symptoms when we're exposed to organism X. So it's a pretty good guess, but it's a guess still. And we're probably, all of us are aware of situations where a physician or a healthcare provider has been wrong about a diagnosis, in particular an infectious diagnosis or an infective diagnosis. And so this is some of the challenges associated with the field. But the thing to point out is that microorganisms are ubiquitous. They're everywhere. This room is loaded with different species of each of the groups of microorganisms. We'll even have some of the algae that are in here. And they're going to be obviously associated with doors and windows, areas where they have access to natural light since they photosynthesize. But everything is, this podium is coated, your chair is coated, your skin is loaded, your clothes, your paper, your food, everything. So microorganisms are everywhere. And this was something that was not appreciated when I first started off as a microbiology student. And that idea persisted up into graduate school. But as a microbial ecologist, we were constantly going, this doesn't make any sense. Yeah, they have to be everywhere. Why wouldn't they be everywhere? That was the question, not justifying why they would be, but challenging everybody to answer the question, why wouldn't they be? Because they have the ability to survive in a broad spectrum of environments. And it turns out that we are heavily dependent on all of these groups of microorganisms, which now provides the foundation to the field of microbial ecology. We now know, when I talk to my medical microbiology colleagues now, and some of them are still very focused on organism X causing disease Y, and that is crucial to the field. That's crucial to us as humans. We need that type of research being done. But I now have medical microbiology colleagues that are not cause-effect, not this organism causes that. Instead, they're actually starting to look at the microbial ecology of the human system. As we, as animals, represent walking incubators for microbes. And why wouldn't we, right? We've got this awesome temperature thing going on inside. We got a lot of sugar and organic carbon for organisms to consume. And look, our fungi like organic carbon, our protozoa like organic carbon. We didn't really talk about it, but so do the bacteria. Yeah, so we create this, we present this great environment for microbial growth. And diverse microbial growth. We have pockets in our bodies and on our bodies that have oxygen. We have other pockets that don't have oxygen. And there's groups of microbes that are adapted to those no oxygen environments. I mean, it's awesome when you really think about it. And now with where we're at, we have a new field called medical microbial ecology. And those individuals are specifically focusing on the microbiome, and a lot of us have heard about that in the press lately, the microbiome of the human system. And when we look at where microorganisms live, everywhere, from the table, the chairs, to the inside of our guts, our mouths, our skin, if we were to take a microscope to each of those environments, we'd see something that looked eerily similar to this. This is called a biofilm. And we now know that the majority of microorganisms want to live as a biofilm. In other words, they don't want to be all by themselves. They want to be together with other types of microorganisms. I got yelled at in my microbiology class because I raised my hand and I had a question. This was like a mega class. There was like 200 students in there. So I was mortified when this happened. I raised my hand and I said something like, well, don't these microbes want to work together? It was actually, don't bacteria want to work together? Because in my head, I was making a connection with human society. And my professor at the time, who was a very traditional microbiologist, yelled at me in front of the entire class and said that was absolutely ridiculous. Because they're all out for themselves. They're not altruistic, like we have a tendency to affiliate with ourselves and with other animal groups. So no, they wouldn't want to be next to each other or near each other. They're competing with each other. That's been totally thrown out the window now. Because now in the field of microbiology, we know that they want to live like this. They want to live together because they're supporting each other. The waste product of one species is food for another. They each have a job to play. Just like in our very own society, we each have an important job to play. And if any one of our jobs goes away as a whole, we have to backfill that hole if our society is to continue to operate the way that we want it to. In microbial ecology, we now believe that that's the case with these different microorganisms as well. And it's not just bacteria working with bacteria, but it's bacteria working with fungi, with protozoans, even with the viruses. What role, I don't think I said this, what role do you think the viruses play in the environment? Not to make us sick, but what do you think they would do when they actually infect other organisms? Natural selection, population control. They're the #1 reason why we're not overrun with, pick your favorite organism. It could be plants, it could be an animal species. Now, there are other factors at play as well, but we've got the viruses. Now, humans, we've removed ourselves from that natural selection. Okay, and we can have a whole discussion about that one too, because I really like that discussion. Okay. So we've sort of removed ourselves from that. But when we look at other groups of organisms, all the way from the microorganisms up to the macroorganisms like the plants and the animals, the viruses play a really important role in population control. And so even in these biofilms on the table and doorknobs and on the surface of your skin, there's groups of viruses at play there as well that are busy trying to control the numbers of different species of organisms. But they're working together in such a way that one doesn't overwhelm the other. So we have this idea of a community or a society amongst microorganisms. Okay, there's your background. So, majority of us probably woke up this morning, well maybe not because it's Saturday. So maybe you woke up yesterday, Friday, right? You got up, you showered, you brushed your teeth, you washed your hands, you maybe microwaved a breakfast burrito. What are you doing with those 4 activities? Why do we shower? Yeah, yeah. We don't want to smell bad, right? Okay. Body odor is caused by different microorganisms that live on different parts of our body. And some of their waste products are different types of organic acids, volatile organic acids that give rise to body odor. So yeah, we indirectly shower because we're microorganisms, because we don't want to smell bad. How about brushing our teeth? Plaque. This image is actually of plaque. This is dental plaque. I took it off of a dentist's website. Okay? This is plaque. Do I want everybody to go like this? Okay? Feel your teeth. And we feel the bumpy, fuzzy, kind of in the layer maybe that right is above the gum line or below the gum line. That's this. If I was to scrape your teeth, take some of that plaque, put it underneath the microscope, we're going to see something very similar to this. Plaque is just a really thick biofilm of different types of microorganisms. So we brush our teeth to get rid of plaque because we don't want cavities and we don't want bad breath, right? So it's a control mechanism. You cook our food. We cook our food. Yeah, because we like to eat warm food. I don't want my breakfast burrito cold. But that's not really the true reason why I microwave my breakfast burrito. I'm cooking it because I don't want to get sick after I eat my breakfast burrito from some foodborne illness. So we cook to kill microorganisms. We refrigerate to control the growth of microorganisms. We freeze so that we can have long-term storage of food products to try to reduce microbial degradation and deterioration of the food. We shrink wrap our food to protect it from the environment so it hopefully either slows down or prevents microbial growth. We use food additives to reduce spoilage of food products. We add salt to our food, not you adding salt to your French fries, but on the manufacturing side of things, we add salt along with those preservatives because salt can be used to control the growth of certain microorganisms. We're adjusting pH. Your potato chip bag is puffed with nitrogen gas because it slows down, believe it or not, your potato chips would eventually deteriorate, especially if they got moist. Okay? So we do all of these things every day, these activities that have become part of just our regular lifestyle. We don't even think about it anymore, the reason why we do it, but we do it to control this. And what's come up since the 70s is in the 70s, the early to mid 70s, we actively started adding additional chemicals to everyday household products to try to increase the antimicrobial properties of those products. That included soaps, detergents, shampoos. Today, it's hand sanitizers. There's a whole family of hand sanitizers that contain a chemical in them called triclosan. Triclosan was really, really popular in the 90s and the early 2000s. And now we know, some of you might have, my son is in Jeffco. Jeffco has now banned triclosan-used hand sanitizers in the classroom because we now know that that antimicrobial chemical, triclosan, which is very effective, by the way, is also detrimental to the human body. And it's been directly linked to a number of cancers. It's been directly linked to disruption of all of the microbes that naturally live on us. And it turns out in us. Because we have a direct relationship with microorganisms. And they're the reasons why we can maintain health, by and large. If we ignore the occasional foodborne pathogen, I'm covered in microbes. I've got a ton of microbes inside of me. Every part of me has a microbial component. We used to think blood was sterile unless you had septicemia. We now know that is not true. There's an inherent microbiome associated with human blood, and these are healthy individuals as far as we can tell. There's now microbiome associated with urine, and we used to think urine was sterile inside of the bladder, at least, before it exited, assuming you didn't have a bladder infection. We now know that that's not the case. We used to think amniotic fluid was sterile. We now know that there's a microbiome associated with amniotic fluid. The list goes on and on. We can't find an environment that is devoid of all microbial life. And when we think about the human perspective of that, we're dependent on these organisms for our health, the human microbiome. But as you start using these antimicrobial products, what are you doing? Exactly. Your intent is to protect, I'm trying to, smear around here on the table, and I picked up influenza on my hands. Now, if I, start sticking my fingers in my nose, because influenza is a respiratory illness, I'm going to inhale some of that virus, and I can develop the flu. So a lot of folks would use a hand sanitizer after touching a surface they suspect might be contaminated with something. Use a hand sanitizer to try to kill off the flu virus. Right? We're just now leaving our flu season. A lot of hand sanitizer was being used the last few months. Right? But as I smear the hand sanitizer around on my hand, we now know, yeah, I might be getting rid of some of the flu virus. That's a big might, by the way. But I'm also, if this represents my healthy flora on my hand, I'm also starting to kill off my healthy ones. And I need these because your skin flora, if we just focus on that, helps modulate skin moisture, skin integrity. The oils that we always thought were associated with the human part of us, we have very, very compelling evidence. It's the microbes that are on our skin that produce skin oils. And skin oils help, keep us looking younger and more youthful, vibrant. So if we start destroying our skin flora, there's a number of consequences that can happen from that. And we now know the release of these types of chemicals, triclosan is just one of them. Think antibiotics for another family of these chemicals. When you're prescribed an antibiotic, you're almost always prescribed it at a dose that is higher than your body can metabolize. Because when you're given an antibiotic, it's a pretty serious situation, or at least ideally it should be. A pretty serious situation. So your healthcare provider isn't going to mess around. They want to get rid of whatever this infectious agent is, and they're hopefully suspecting it's a bacterium. Because antibiotics are really antibacterial chemicals, and they're only effective against the bacteria. They won't touch the other groups. So you're given this chemical to try to rid yourself of the pathogen that's causing an infection. But as you're doing that, that antibiotic gets into your bloodstream. And now it has an effect on your oral flora. And it has an effect on your gut flora. And it has an effect on your urogenital flora. And the list can go on, especially if you're on these chemicals for a long time. And as you're destroying your healthy flora in an attempt to get rid of the pathogenic organism, the health consequences are adding up. So just thinking about humans, because we can all relate to this, we now know that disruption of our healthy microbiome is directly related to illnesses like irritable bowel syndrome, allergies to food, both respiratory allergies as well as foodborne allergies. There's a lot of compelling medical research that's coming out to show that when we disrupt our GI tract flora, it changes behavior, it changes learning. And if this is happening in a child, it changes development. That could lead to permanent long-term modifications. So the list is really intriguing. I just came across a study that is linking the gut microbiome with breast cancer. Because it turns out our gut flora can help modulate hormone levels in the human body. Because our microbiome is cross-talking with the human of us. And the human part of us is cross-talking with the microbiome. But if we don't have messages that are being received and interpreted appropriately, that's when things start to go awry. And it turns out that the human body, we are 90% microbial. If you literally just took me apart and just added every single cell, I would be 90% microbial, 10% human. Now, the numbers have changed a little bit. We're looking at, maybe 10 to 30% human. We're still trying to figure that out because we don't know what to do with some of the ones that are just sort of loosely associated with us. But it doesn't matter how you look at it. We're a significant microbial ecosystem as individuals. So where do we go with this whole idea of chemical disruption and how organisms interact with their environment? Okay, so human microbiome is part of this story, but the other part of this story is knowing that microorganisms play a role in, for example, agriculture. Our ability to grow the food that we can grow, and this is global, is heavily dependent on the different species of microorganisms that are present in soils. All of our aquatic environments have their own set of microbiomes, if you will. And those are important to also helping sustain plants, as well as animals. All of us as animals get our microbiome, the initial inoculum, from our environment. As humans, we get our initial inoculum from the vaginal canal during a vaginal birth. One of the reasons why C-section babies sometimes have a propensity to develop certain types of illnesses, because they're not picking up mom's vaginal flora as they're passing through the birth canal. So we get it from our environment, though, whether it's from each other, the environment, from soil, from water. Same thing with plants and other animals. So now you have to ask yourself, if the chemicals that cause disruption in us, such as triclosan and overuse of things like antibiotics, if they cause disruption in us, what does that mean in terms of the environment? And that's really where I'm interested in. So what happens to the ability of these microorganisms to degrade organic carbon in the environment? What happens to their ability to cycle things like nitrogen? I don't know how many of you might know about the nitrogen cycle. But the majority of nitrogen, which is a crucial macronutrient for all biological systems, majority of it is in the atmosphere. It's as nitrogen gas. We can't use nitrogen gas. Plants can't use nitrogen gas. The only organisms that can use nitrogen gas are 5 species of microorganisms that we've identified so far. And they're mostly bacterial. 5. And it turns out they're also extremely sensitive to antibiotics and triclosan and other suites of chemicals that have antimicrobial properties that we're releasing into the environment. So the macroecologists suspect that if we continue on the trend that we currently are, and they're monitoring now the best as they can these global levels of these nitrogen fixing bacteria that can take gaseous nitrogen and they fix it into ammonia. Now ammonia is what many biological systems can use. Not exclusively, but most. And if those groups of organisms die off because we have an increasing amount of these chemicals that we're releasing into the soil and water globally, they predict the entire nitrogen cycle collapses. Because when nitrogen dead ends, it dead ends in the atmosphere. If we don't have a way of pulling it back out into biology, then biology doesn't have access to it. And the ecologists are predicting worldwide collapse when that happens. So they're looking now at rates of loss of these certain groups of microorganisms. In microbial ecology, we almost never say definitely. It is looking very likely, if we continue on the trend that we're on, that it will happen. But I'm going to give you perhaps something that might be the saving aspect of all of this, right? So, as we lose these groups of organisms, this is where we're at in microbial ecology, is trying to ask the question, what's going to be the consequence? We got the medical folks that are looking at the human system. We've got some veterinary folks that are actually looking at other animal systems. But what's going to happen globally? And so here's one last comment, and then I want to open it up to questions, because I usually get a lot of really fun and awesome questions. We've identified, of all of the groups that I had talked about, if you added them all up together, we've identified at most from 1 to 10% of what's out there. That means that we have 90 to 99% of microorganisms that are out there in our environment. And that I'm going to include, inside of us as well, us representing another environment. That means that we don't know who or what or where 90 to 99% of the microorganisms are, what they're doing, and where they exist. And one of the reasons we have that limitation goes back to this need of genetics. Because we can't just look at them and go, ha ha, you are, and then fill in the blank. We have to go, wow, okay, I have no idea who you are. So let me collect some of you, extract your DNA, sequence your DNA, and then compare your DNA against all of the others that we've identified. And if you match, we know who you are. You don't match, we don't know who you are. Then we have to go through a whole lot of work to try to grow the organism in the laboratory, try to give it an identity, and then in the process of doing that, trying to attribute function, characteristics, behavior. So if we don't know who or what 90 to 99% of the organisms are doing in the environment, and yet we're indiscriminately using these chemicals that target pathogenic organisms and non-pathogenic organisms, ones that cause disease and ones that don't. From an environmental standpoint, as we're killing off organisms, we have no idea we're even killing off. What's going to be the global consequence? I'll leave it at that. Thank you so much for listening, and I'm happy to answer any questions. Yeah. Oh.
Gut biome and crosstalk. I was just wondering if you had some other way to describe that a little bit better.
In terms of how the two communicate with each other?
Yeah, because we, as you say, they're single cell. They don't have vocal cords, and to talk means you got to have a brain, and somebody's got to be receiving it. What do you really mean?
Okay, so thank you for that question. When we talk about either microorganisms talking to each other or talking with other biological systems, including our own cells, what we're talking about are chemical signals that are being transmitted back and forth. And so it's the question of, is my gut flora as a collective releasing chemical signal X that then my cells are picking up, and in response to that message, my cells are going to do Y. And if chemical X is missing, then we've got a broken link. We don't have the crosstalk. So it's chemical signaling. So if keeping that in mind, you can imagine that if you have a system like this and you have a disrupted microbiome, so some critical members missing, and chemical exposure is one of those causes. If that happens to be the same species that's responsible for producing chemical X, then we're missing that message. So then what's the consequence, right? Am I following you with the, okay. He's in charge.
Thank you.
Yeah.
So just a quick question. Probiotics, the kind that you buy in the store,
Yeah.
good or bad?
Okay, I'm going to give you my opinion.
Please.
Okay, I'm getting on my soapbox. I have a lot of soapboxes, so this is one of them with the whole probiotic thing. Okay, so let's think about, because the majority of us are interested in probiotics to try to keep a healthy GI tract. We have this idea that we have a lot of microbes living in our gut, we want to keep it healthy, or we're trying to replace flora because we were just recently sick, you had a lot of diarrhea. When you have that gastrointestinal upset, it flushes a lot of your gut flora out. Not entirely, but it can have an impact. Okay. Probiotics have become a lot more sophisticated than they were when they first hit the market. When they first hit the market, it was dominated. If you bought your probiotic, whatever it is, and it doesn't matter if it's like a refrigerated version or in your yogurt or dehydrated that you're getting off of the shelf at a natural food store. So wherever you're getting your probiotic, it started off that they were dominated by one or two species in your probiotic. Lactobacillus is one really common with. Bifidobacterium is another one we're really familiar with. Now, when you buy a probiotic, especially if it's a more expensive one, I'm not saying high-end, just saying expensive. The list is a little bit longer. But let's say, let's be generous with it and say the list has 10 different species that are in there. The reality is when we look at the gut microbiome, the studies that have been done have identified thousands of different species that make up a healthy human gut microbiome. So the reality is, what are the odds that the 10 that were chosen out of the thousands that you need are really important at that moment in time? So that's a question we haven't been able to clearly answer. The other part of it is this. What we now know about the human gut microbiome if you think of the gut as a tube, just like a pipe, okay? And so in the middle of the pipe is where it's open. Just like water would go through a water line, your GI tract is a pipe, your food and everything passes through the center of the pipe. The lining of the pipe on the inside, that's where your gut flora is. And it could be actually millimeters in diameter, because our gut flora is a very prolific biofilm, microbial biofilm. So you've got this really thick layer of microorganisms attached to your GI tract itself, and the food passes through the middle. Okay, everybody have that? Okay, so when you take a probiotic, those microorganisms pass through the middle. And it's really hard for them to make their way into what can be easily millimeters thick biofilm to be able to stay in place to have, hopefully, a positive impact. So the aspect of retention of probiotics is also something that's very controversial. So in my opinion, do they do any harm? Absolutely not. They're a great source of vitamins, because microbial cells are really easy for us, our bodies, to degrade. They start falling apart in the GI tract. That's a whole other part of probiotics too. Not all those organisms survive stomach acidity. Okay, so I don't think they're harmful, but do they have the intended benefit? That I'm not convinced of. What I am convinced of, if you want a true probiotic, is fresh fruits and vegetables. Because they're uncooked, so you're not killing a lot of the naturally occurring microbes that are on fresh fruits and vegetables. And if we, even if you wash them in the kitchen, you're getting rid of the things that are loosely associated, but you're not getting rid of the organisms that are more tightly bound. So those organisms, because they represent diversity, because they represent our environment, where we naturally get our microorganisms, those are more likely to be a more effective probiotic. Okay. Oh.
Question over here.
Hi. So I've read articles and theories that where there's one theory that the primary cause of autoimmune diseases is all this sanitation and all these things that we're constantly doing. I was just kind of wondering your thoughts on that.
I certainly wouldn't put, and I'm not an immunologist, although I like having arguments with immunologists. From an ecology standpoint, there's several over at the Anschutz Medical Campus that I'm good friends with, and we have some really great discussions about, are we tipping human ecology with vaccinations and medicines and so forth, because we've removed ourselves from the natural selection process. But that's a separate discussion. But as far as autoimmunity, I certainly wouldn't say that the majority of autoimmune disorders are caused by something with a microbiome. However, there are several known autoimmune disorders that are caused by either a disrupted microbiome, and actually attention deficit disorder in some kids. They think this is going to be a pretty complex disorder, just like you're not going to be able to pinpoint one cause of all cancers. But one of has quite a bit of evidence to show that these kids don't have a healthy microbiome. Some of the cell signaling, the communication, isn't going on back and forth between the microbiome and the child, the human part. And because of that, it's modulating different types of endocrine levels in the human body, and then that in and of itself is having some neurological impact. And in part of that is that the immune system can also be trained to see something it shouldn't normally be seeing when you have a disrupted microbiome. There's other cases of where, for example, some forms of heart disease is actually caused by an autoimmune reaction on part of the body. And if individuals with strep, for example, that had some really severe strep experiences, either as children or later on as adults, there's a protein on the surface of the strep bacterium, the Streptococcus bacterium, that is also very similar to a protein that's on cardiac tissue. So your body, in attacking the Streptococcus bacterium, mounts an immune response. It develops memory to that protein, which then all of a sudden it starts recognizing and seeing on the surface of cardiac tissue. So it'll be an autoimmune degradation of the heart muscle that can then lead to weakening of the heart and heart attack. So I think there's definitely a link, I think in some cases, and certainly the idea of allergies has an immune component, and we know that a disrupted microbiome, in particular a gut microbiome, has been linked to some types of respiratory allergies as well as foodborne allergies. I think there's a lot left to be discovered about where the immune system comes in and plays a role in this whole story.
So on a slightly lighter note, are you saying the 5-second rule's not...
Yeah, we have this discussion in my general microbiology class every semester. No, I'm not worried about the 5-second rule. Now, if it rolls around in that 5 seconds and there's a bunch of stuff attached to the food, there's just a gross factor that's involved, okay? Whether I got to worry about the microbes or not. And as my son was growing up, my initial reaction as a microbiologist was don't let him eat anything that had hit the floor. But then the microbial ecologist would come out and I'd be like, he's got to eat stuff that hit the floor. So I have to kind of do this sort of thing. And I just tell him to hurry up and do it. And that was my attempt to try to boost his immune system and his tolerance, yeah. Yeah.
So I wake up in the morning, I have a big old latte with a bunch of cream and a bunch of sugar in it and a bunch of caffeine in it, which I don't think my gut evolved to handle.
Okay.
Is my gut happy or is my gut sad? And if I can do one more question, if you can speak to artificial intelligence in research on here, that'd be great.
So, so when we think about things like lactose intolerance, right, which is on the rise. Now, with a lot of these food allergies that we have, the idea, it's one of two, So with a lot of the food allergies, lactose in particular, my husband's lactose intolerant, and he's not here, so I can talk about him. So he's lactose intolerant, and we all, I think, know what happens when somebody who's lactose intolerant consumes lactose. There's a lot of GI tract upset. He wasn't born this way. I mean, it happened when he was a young adult, and it happened after a fairly prolonged antibiotic regime. And this has been known or shown to be the link in some individuals. So if you're on a long-term antibiotic for whatever the reason is, what you're doing is that antibiotic is targeting some of your healthy microbes, as well as potentially, hopefully, right, the pathogenic ones. And so if you lose some of those organisms that would otherwise, in this case, maybe degrade lactose, you don't have them present anymore. Oh, okay. So let me. Do you grow bad bacteria in your gut by eating the wrong foods? Okay. There are no bad bacteria, so I'll just. Okay. That's why I stuttered a bit. I was like, oh, wait a minute, what are we talking about here? Thank you very much. Yeah. No. Okay. All right. So yes, you definitely modulate your gut flora based off of your diet. Absolutely. So if you eat a lot of simple sugars, okay, candy bars, sweets, adding a bunch of sugar to your coffee on a regular basis throughout the day, what that does is it starts selecting for the microorganisms in your gut that are going to prefer that as a food source. And if you're eliminating other potential sources of food for other groups of organisms, then those will start to die off.
But there's no such thing as bacteria in your gut.
There are pathogens, absolutely, but not pathogens.
Bad effect though if it gets lopsided, right? If you start getting one and not the other.
Well, that's the thing, yeah. So would I say that they're bad bacteria, ignoring the pathogens? No, I wouldn't. But when you change your gut flora, yes. Now you can have problems if you're eating different types of foods, so it could cause gastrointestinal upset, or you're not getting the chemical signaling that you need because some of the ones that died off were the ones that are going to produce chemical X that your body needs so it knows when to produce chemical Y.
I mean, we, as humans, we consume chemically way more than we evolved for.
And I throw fake sugar.
And fake sugar, okay. So there's research out there that shows that different groups of microorganisms can degrade even the fake sugars. They're different than the ones that will go after the real sugar, for example, glucose and sucrose and so forth. We don't know enough, though, to be honest, about all of the potentially thousands of different members of the gut flora. And that's just, by the way, thousands that we can identify. And if we're only identifying 1 to 10%, that tells you we're easily missing the majority. So of the ones we've identified, we don't necessarily know their role in every case. We might know that they like sugar or like fat, like proteins, for example. But within that context, what is species X actually providing for that microbial society? And then also, what is it providing from the human standpoint? Okay? So that was a roundabout. But let me add to that really quick. They now can tell by just taking a fecal sample, and if they know nothing else about you, they can tell whether you're vegetarian, whether you eat meat, and about how much meat. If you're heavy on the meat side and you're not eating a lot of fruits and vegetables, your gut microbiome will have a very characteristic fingerprint or a profile. If you're vegetarian, your gut flora will have a very characteristic profile. And if you're in between, it's kind of a merger in between. And thinner individuals actually have a characteristic gut flora than individuals that might otherwise be overweight as well. So they're looking at the fingerprint of the gut flora to try to use that as a predictive mechanism of the person. Personalized medicine, this is one aspect of it. Is can we look at we're really, really, really far off. But can we look at an organism's microbes, skin, gut, mouth, potentially, right? You can go through the list and then make assumptions or conclusions about the behavior, the health, diet, exposure of that individual. So that kind of gets to that as well. Yeah.
Mike, that was just mentioned there. You gave us reason to be skeptical of orally introduced probiotics or biotic organisms. What about the fecal transplant?
Yes. Thank you. Have you guys had a talk on fecal transplants? Does everybody know what they are? Should I review what they are really quick? Okay. All right. So individuals with Crohn's disease, for example. So that's a pretty severe gastrointestinal disorder. And it can be to the point where it's debilitating for these individuals because they will have regular, frequent, and severe bouts of diarrhea, gastrointestinal upset. And it can get to the point where they basically can't, it's difficult for them to hold down a job because they always need to be near a restroom. So the disease can get so severe. There's been a lot of research that's been put into causes of Crohn's disease and then ultimately how to treat it. So here's a way cool factor with Crohn's disease. They've now been able to associate Crohn's disease with the absence of specific species of bacteria. So they now know that these individuals are lacking certain members of their gut flora that need to be there, that are basically going to tell the gastrointestinal tract, stay calm. Because if you think about the GI tract, it's a very abrasive environment, right? So think, if you eat a salad, we all eat salad because it's roughage, right? What does roughage really mean? Well, it's something that doesn't easily degrade through the GI tract. So it kind of scrapes the inside of the GI tract. It's this big, bulky thing. It might have edges or corners to it. It's got a lot of structure to it. So the idea is that it cleans out the GI tract. If you just think about the food that you eat on a regular basis and the fact your GI tract is a muscle, it's a series of muscles that are constantly contracting and relaxing. So it sort of moves in waves. So there's a lot of potential for tearing of tissue lining the GI tract. And when you have a tear, your immune system responds as it should, right? And it basically, because there's going to be inflammation because of an immune response, because your immune system's going, there might be something wrong here, let's go and protect the area and figure it out. One of the consequences of inflammation in the GI tract is diarrhea. Individuals with Crohn's disease, especially severe Crohn's disease, have basically chronic inflammation of the GI tract. The species that they're missing, we've been able to attribute function to them. And it turns out they produce a natural anti-inflammatory. So they're there, and they're supposed to be, if you imagine, go back to the GI tract as the pipe. So you have the pipe part. These organisms that produce these natural anti-inflammatories are supposed to be sitting right on top of the pipe, right on top of your GI tract. And it's a waste product for them. So they're always producing this anti-inflammatory. So in healthy individuals that have a high abundance of these species, that keeps the inflammation down. Individuals who are missing these, the inflammation goes up because they don't have that natural anti-inflammatory. So one of the treatment methods, because it's very difficult, if you're missing a group of organisms that produces the natural anti-inflammatory, but you have all these other organisms that are stacked on top of it, right, the biofilm. It's very, very difficult, if not impossible, to get the needed organism through the biofilm all the way down to the bottom and get them to grow in the numbers that are needed that will coat the GI tract. So the idea with fecal transplantation is that they'll go in, they'll take an individual, they'll give them high dose antibiotics. It's a whole suite of antibiotics, very high dose, and it'll be prolonged. And the idea is to kill off as much as possible in that person's GI tract. Then replace it with organisms that are coming from the gut of a healthy individual. Fecal material was the original source for that. So originally when this technology was developed, they took fecal material, bowel movements from healthy individuals, and they used that, put it in a slurry, and used it, they delivered it via an enema, but they used it then to provide that diverse inoculum to repopulate the GI tract. So fecal transplantation, taking fecal material from one person and using it as an inoculum for another person. And it's been wildly successful. And now what they're trying to do is to figure out all of the critical organisms and put it they have actually dehydrated fecal material, to put it bluntly, which is very similar to what the wastewater treatment industries use, sometimes to re-inoculate the wastewater treatment process. If they have a chemical that comes through the treatment plant, wipes out their organisms that are busy cleaning up that water, they can buy these dehydrated pellets almost of basically it's fecal material, because that's the primary inoculum for a wastewater treatment plant. So it's a very similar approach with the fecal transplantation now, is they've got this dehydrated version. There's a lot of work though trying to really refine that and elucidate exactly which species are needed. So it's a really cool technology. Oh, wait, right behind you. Yeah. He's got the microphone.
Yeah,
so.
I got the mic, so I'll pass it to you afterwards. Most of the pharmacologic antimicrobials we have their origin in a natural source.
Yes.
And in most of these, if you will, films, there's a chemical warfare that's going on all the time. And I understand that if we look at our human ecology, when we take high doses of antibiotics, that we get quite disrupted. But what is happening to the overall environment? Is it a matter of dosage of the amount of antibiotics, antimicrobials that's getting into the environment that is the issue?
Yes.
As opposed to what's happening at the microscopic level?
Yeah, it's a discussion of scale.
What environment? You're talking about inside your body or like the environment?
Anything and
outside.
But even inside of your body, right? Some of the same. So what the question or comment had to do with is that when we look at antibacterials in particular, if we just focus on that, which is a subclass of antibiotics, if we just focus on the antibacterials, 90% of them are actually produced by other microorganisms. So penicillin, for example, which is our first widely used antibacterial, was discovered by Alexander Fleming, who was an early microbiologist looking at basically fungi. He was actually studying bacteria, and he isolated fungi by accident. And it was the fungus Penicillium. Now, Penicillium is probably growing in all of your refrigerators at home. It for sure is in mine, because I'm not good at cleaning out my refrigerator. My excuse is I'm a microbiologist, so I want to see what grows. Okay, that's my excuse. But, so I got a lot of fuzzy stuff growing on things in my refrigerator. Some of that is bound to be Penicillium. So it's a very prolific, very common organism. But penicillin comes from a fungus, and a lot of our antibacterials are coming from other microorganisms, such as the fungi. So the comment or the question is, if we realize that these fungi that produce these antibacterials are naturally occurring in the environment, some of which also occur in our GI tracts, and they're always sort of producing these antibacterials, what are we really doing when we take an antibiotic, an antibacterial that's been prescribed? So in other words, how are we changing the system if these chemicals are already out there? And it's easily a matter of scale. It's magnitude. Because in nature, if you imagine, if this is a bacterial cell, you know, the average size of a bacterium is 1 to 2 micrometers. And if I'm a hyphae, which is a single cell of Penicillium, and I'm next to this bacterium, I'm producing penicillin, but it's only impacting this right around me. It's not actually accumulating at least at detectable levels that we can detect so far analytically in the environment. So it's happening at this very micro scale in levels that we can't even measure. But certainly those levels are affecting other microorganisms. So now when we come in and you have a human that's excreting an antibiotic because the body hasn't fully metabolized it, it's coming out in magnitudes higher than what would be occurring in nature. So antibiotic resistance is not a human-driven process. The commonality of antibiotic resistance and the spread of it, that's being human facilitated because of our use of antibiotics.
But is the dosage of human use, can you not hear me?
I can hear you, but I don't know about the rest of the group.
Is the dosage involved in human use identifiable in the overall environment at levels that nitrogen fixing and so on organisms are being impacted by the human initiated dosage?
Yes. So far, the studies that have been done looking at these more global impacts of antibiotics and other antimicrobials, we're measuring those chemicals at levels that are going to be human associated based off of our use, and we're seeing the impact. We can't clearly define whether that impact would still be there because of naturally occurring, if you will, not human intervention, but naturally occurring levels of at least the antibacterials. So would the nitrogen cycle, in other words, still be falling apart, so to speak, if we removed the human contribution of the equation? I mean, the ecologists are going to say no, because the levels are too low. We're not causing this massive die-off of organisms. Okay. Sure. Yes.
For the group at large, you know this, but if you add up the number of bacteria in the world, it's about 10 to the 30th, 10 to the 31st. You put them end to end, and it's 100 billion light years. And that doesn't include viruses, which is about the same number. So you get that massive amount of bacteria, which weighs about the same as the United Kingdom, which is known.
I'm going to add to that your numbers, by the way, but yeah.
And you, this Darwin guy says they're mutating and replicating. I believe some of that, not all of it. How come none of them seem to mutate to the point that they kill us all? They should, but they don't.
Okay.
The numbers say we got to be the luckiest things in the universe.
Okay.
I think something else is going on, but I'm curious to hear your take on this.
Yeah, absolutely, and it's a great question. So I'm going to add to the numbers you were putting off with the number of bacteria. Okay, so what the question is basically we have a lot of bacteria in the environment, all of the groups of microorganisms in the environment. They far exceed the numbers that we see with any other biological system. Insects, animals, plants. I mean, they're at numbers that we even have a difficult time really comprehending as humans. And the question is, why are we not, basically, why are we still here? Why haven't we been wiped out, whether it's by a group of microorganisms or by a single organism? In other words, why haven't they evolved to just take out the humans? Okay.
And everything else.
And everything else, and everything else. As an ecologist, that doesn't make sense. If we have microbes that want to live in us, that prefer, let's say, our GI tract because it's the right temperature, it's got the right nutrient loads, there's other chemicals that our bodies produce that actually help support and feed these microbes. If we think about the fact that these microbes have found niches where they like to live, including us, they wouldn't want to wipe us out. That wouldn't be the objective. One of the reasons why pathogens in general, when you look at them longer term, now we can argue what longer term is, that's sort of the discussion right now, why they don't increasingly become pathogenic and get to the point where they will kill us, is because that's counterproductive. If they need us, they don't want to kill us. So HIV, for example, has been getting less and less effective and infective as a virus. So individuals, even in the absence of medical intervention, individuals with HIV are actually living longer and longer when we look at HIV individuals in other parts of the world. It's still a deadly disease. But the virus itself is losing some of its pathogenicity, which is what the ecologists would predict. They predict that at some point HIV will have, harmonious is not maybe the right word, but a more balanced relationship with humans. Cold virus, they think, used to be a lot more severe than in general what it is. Influenza, we know, used to be more severe. Now influenza is an interesting story because we have new strains that are constantly popping into the human population. Okay, but the idea is that if you have a pathogenic organism, it wants to make you, not that it wants to make you sick, it finds you an ideal environment. Your body reacts to it. That's ultimately what makes you sick, because it'll infect certain tissues or live in certain tissues and your immune system will respond from an ecological standpoint. But it doesn't want to get to the point where it will kill you. Ebola, for example, is a virus that we think is fairly early in its evolution in terms of its relationship with humans, because it'll kill you and it'll kill you quickly. It has a very high mortality rate. The ecologists predict, especially the viral ecologists, predict that Ebola will become less and less severe. The infections will become less and less severe with time, once that virus figures out sort of a balance. Because for Ebola, it doesn't want to kill its host, because that means it also can't be spread as easily. And our outbreaks so far have been, compared to outbreaks, relatively contained in part because of that.
Is that just because the bacteria are getting smarter at fighting off viruses, and less because the viruses want to be nice?
It's not necessarily that the viruses want to be nice, right? We can argue about whether they really have a decision to make in this process, but mutations are happening all the time. And some of those mutations can change behavior. And so overall, if a virus mutates in such a way that it kills its host more quickly, then it's going to be isolated to a smaller outbreak. But if a virus mutates in a way that it makes you sick and you're sneezing and you're coughing, but you're spreading it, right? Now it's survival of the species as opposed to survival of the individual. And it makes more sense from an ecological standpoint to spread into many more individuals. So you cause a less severe virus so you don't kill your host, but your host has more opportunity to spread. That's one of the ideas behind viral evolution in plants and in animals, actually. So it's really an interesting discussion. So it kind of gets at maybe what you were asking.
So they don't want to kill us. They're our friends.
No, I wouldn't say they're our friends. And that's part of what we have to do, right? Is we have to break ourselves of thinking of these organisms as friends or enemies, right? We're just habitats. That's all we are, is just habitats. And sometimes when they get in us, they make us sick. Right? Again, the ecological perspective. Where'd the microphone go? Yeah.
So you talked about hand sanitizers. Talk into it. You talked about hand sanitizers, so I am trying to make a switch, but I don't know what to switch to. So what do you use?
I use soap and water.
Just like Ivory soap or?
No antibacterial. So one of the reasons I know this is because the lab I was in as a PhD student, and I cannot reveal the company's name. But one of my fellow PhD students, I was not on this project, but her project was to look at the effectiveness of these antimicrobials that were being put at the time in some very high-profile commercial products. And her job, she was sponsored by the company, so her research was sponsored by one of these companies. They wanted her to test the effectiveness as a third-party entity to test the effectiveness of these antimicrobials against soap and water and just water. And so she had a whole suite of things she was testing. They weren't any more effective. And as a matter of fact, in many cases, they were less effective. Because one of the things that we do with the hand sanitizers in particular, right, is you go squirt, squirt, squirt. So I rub the palm of my hand. That's what most people do, is maybe they do the back a little bit. They don't get in between the fingers, jewelry, nails, up on the wrists, right? So one, there's a human behavioral component that's limiting the effectiveness of these chemicals because we're not putting them all over. The other part of it is this. So let's say I touched a surface that has, let's pick on influenza since we were just talking about it. I touch a surface that has influenza, I have it on my hands, squirt hand sanitizers, and I do this. Did the influenza go away? No, still there. I haven't physically removed it. So if I haven't gotten the hand sanitizer effectively on all of the influenza that might be on my hands, then I still am carrying viable influenza that could easily get into my respiratory tract and cause an infection. Whereas good old-fashioned soap and water, you have soap, which is just as effective at killing as triclosan, for example. Studies have been shown that. And you have the mechanical removal of the water and the hands. So soap and water are much more effective. Now, you're not always near soap and water. And in that case, I will use just water. I avoid nobody in my family uses hand sanitizers because there are some secondary health effects to the exposure to the chemicals. FDA is currently actually looking at alcohol. Now, alcohol we don't typically associate. We associate it with cytotoxicity. That's why we use it. It's a very effective disinfectant. We use it in my lab. We use it in the research lab, the teaching lab. But they're looking at potential health effects from not so much the ethanol, but all the other chemicals that are needed to keep the ethanol soluble in the gel that makes up these hand sanitizers. So that brings up actually a separate discussion. Sometimes it's not the antimicrobial directly, but some of the other chemicals that are needed to stabilize it. Oh no, liquid soap is fine. Bar soap, liquid soap. Yeah, because soap is really good at disrupting membranes. And when you disrupt an organism's membrane, it doesn't stand a chance. We don't have resistance against membrane disruption that soaps cause. We do have resistance to many of the chemicals in hand sanitizers, by the way, including triclosan. Yeah.
Switching to the topic of industrial waste cleanup. So my understanding is that we are using less chemicals than we used to, like in the 70s, and they're less toxic, and that's just because of strains and new developments and how we do agriculture. And so I wanted you to touch on that, and then because you're talking about the problem of it, and so is it less of a problem, I guess is where I want to get. And then more, how are we proactively using microbials to clean up either industry? Is it getting better? Is it a solution to some of this stuff, from agriculture to mines and whatever other chemical manufacturing we do?
Okay, so the first part of the question is we are reducing the amount of chemicals. If you just look at volume, or mass, concentration, but we're diversifying our chemicals. So there's constantly new products coming out that are commercially available for agriculture, for individual home gardening, for medical reasons. So we're diversifying the chemicals. So while we might have reduced our use of some chemicals, we're increasing the use of our chemicals in general, as a whole, as a society. That hasn't changed, okay? So when these chemicals get released, and when a new product comes on the market, like a new medication, the pharmaceutical industry seems like it's a regular introduction of different types of chemicals, right? And we're not going to talk about whether they're needed or not needed, but when these things get released into the environment, we don't know what the environmental consequences are, because that's not part of the approval process for a drug. There's the human part of it, the clinical trials, the toxicity assays, etc. But once that chemical, which it will, it's not a matter of if, it's just a matter of when, you have a large enough enough people using that chemical, and it gets excreted in high enough concentrations into the environment, what impact is that going to have in the environment? And that's not part of the drug approval process. That's part of other chemicals. Sometimes the agricultural chemicals have to go in and do ecotoxicity assays. But the majority of our chemicals don't have to go through that process. So we don't know. From an ecology standpoint, and certainly from a microbiology standpoint, it's very difficult for us to predict what the impact of chemical X as a new chemical that we may not have seen before is going to have on the ecosystem and the microorganisms as a whole. We can try to predict it based off of chemical structures and classes of chemicals. Sometimes mode of action can give us some informative information, but we just don't know. So it's a big black box. So it's kind of almost all right, well, it's being released and we got to see what happens. The big class of chemicals right now that we don't know what's going to happen from a microbial standpoint, but also easily we could argue we don't know what's going to happen from an animal or a plant standpoint, are the personal care pharmaceutical products. And EPA is currently not regulating these products because we don't know what the concentration is that's toxic in the environment. We're just now figuring out that estrogen, when you have hormone replacement therapy, that the hormones are given to women in excess of what the bodies can metabolize. They excrete these hormones in urine at levels that far exceed what would happen if you had a woman who wasn't taking hormone replacement therapy. And we're just now starting to figure out, well, what impact does estrogen and progesterone have on animals, which we've long suspected is linked to feminization and decreased reproductive rates and so forth, fecundity. But we don't know at all from a microbial standpoint. So when we look at use of microorganisms for chemical detoxification and degradation, which is my particular area of interest, it's almost like reinventing the wheel every single time. We'll have chemical X that's really of interest for whatever reason, and usually that has to be some larger like health impact or animal impact, fish dying or something to that effect. We go, okay, we got to take a look at this. And then it's almost like, well, we don't know what's going to happen. So we sort of start from ground zero looking at whether microorganisms can help us degrade that product or not. Are there cases where they can? Absolutely. Over and over and over again, it's been shown that especially the bacteria, but the fungi as well, are quite prolific at adapting to different types of chemicals in their environment. And they'll switch their metabolisms in such a way that they'll start using those chemicals as a food source. A pharmaceutical is a form of organic carbon. The fungi love organic carbon. In nature, organic carbon is primarily occurring in the form of cellulose, chitin, for example, which causes that hard exoskeleton on insects. So they can make that adaptation to degrading something else that we consider a chemical. But can they do it at the rates that match what we're introducing into the environment? And that right now is not happening. So when we look at remediation and using microorganisms for remediation, one of the things I have to look at is, are there organisms capable of degrading chemical X or pharmaceutical X, whatever it is? And then when the answer to that is yes, which it invariably is, then I have to say, what do I need to do to change the environment in such a way that I can get better degradation rates? Do I have to increase the amount of oxygen that's available to those microbes? Do I have to give them more nitrogen? Maybe there's not enough nitrogen in that soil. So now I'm looking at environmental modification. And when you talk to the average person on the street, and you say, I'm going to modify the environment so I can increase the growth of microorganisms, then we have a problem. And one of the other aspects, challenges to the field of microbial ecology, especially environmental cleanup of chemicals, is perception. We have a huge PR problem. And it's hard to overcome the PR problem when people are largely driven by fear of pathogens. No, because our federal agencies won't let me. Because if I'm going to treat an environment, clean up an environment, I fall under now EPA. And EPA will be watching me. And EPA has very specific guidelines that prevent me from doing some of the things that would otherwise clean up the environment. In part, I'm not faulting anybody, but it comes down to also PR, funding, politicians, regulators. And sort of they too have some of the same fears that somebody in the general public might have. So the science hasn't quite convinced people yet that it's a useful approach. Yeah.
Getting back to probiotics, I have just a couple questions. Take them at all or no? And if you do, if you should take them, they've got pills everywhere from 1 billion to 75 billion. What's the right, amount to take? And then the other question, you had mentioned something about nitrogen, and you said there was a solution, but I don't know if I heard the solution, about the nitrogen problem.
So to the nitrogen problem, we don't have a solution yet. So one of the things we're really worried about is the majority of nitrogen, when you look at the nitrogen cycle, it sort of dead ends in the atmosphere. So when you degrade, for example, when a plant dies and it gets degraded to the point that it doesn't look like a plant anymore, the nitrogen that might be associated with that plant material when it was alive will eventually get released into the atmosphere. It's a process called denitrification. And when that nitrogen becomes a gas, now it's not biologically available. So we rely on this group, this very limited group, there's about a half dozen that have been identified, that can take atmospheric nitrogen and convert it into ammonia. Now that nitrogen as ammonia is available to biological systems, plants, animals, microbes. Without that critical step, the whole nitrogen cycle collapses. And we don't have a solution yet. So we don't have a fix to that. To get to your other question about probiotics again, when you look at a probiotic bottle, there's all sorts of number of active units, which is what they like to report. Now, probiotics is an unregulated industry at this point. FDA is currently looking at starting to regulate it because they are becoming so prolific. But right now, they're still unregulated. Yeah, they're not regulated because they do fall under the category of supplements. And vitamins themselves are not regulated either. Okay? So from that standpoint, when you say, let's just simplify it, there's 1 million active units of, and then fill in the blank for the organism's name, there's no guarantee you're really getting 1 million active units, number one. And I've done my own, because I have a soapbox on probiotics, I've done my own studies in the lab. I've never been able to achieve what they market. And I'm using the same media that the industry uses, the whole shebang. I've never been able to reach the levels that they're marketing. But the second component to that too is even let's say it is 1 million units and there's 1 million active units that are in there, will those active units all survive before they hit your GI tract? And not all probiotics contain microbes that are adapted to surviving stomach acidity. So now we start getting into how they encapsulate the organisms to protect them, to survive stomach acidity, so that once they get into the GI tract, they theoretically can become part of the GI tract. Okay? So there's a huge question mark, and it's, it's part of the complicated story behind probiotics and whether the commercially available probiotics. And I do not. But I eat a lot of fruits and vegetables, and those are yeah. I was going to tell you what I used to do as a kid, but those are my probiotics. I know I have some medical folks in here, so I know there's a medical name for this condition, but I used to eat dirt as a kid, and a lot of animals eat dirt. Pike, thank you. And so I don't need more. Just saying. If you ever see me, though, out in the garden behind the building, just ignore me. No. But, you know, animals, I think, do it actually, and so they think humans, in part, do it because of they're trying to get a certain microbe introduced into the gut. So they used to say it was nutritional deficiency. Now there's that microbial link that interfaces between the two. He's got the microphone. Pica still exists. Because and those, if there's, right, any medical focus, because why does pica exist? Why is it common during pregnancy? So, you know, at least the temptation to eat soil. It I think is going to come down to a nutritional deficiency, and our microbes are kind of that interface. Yeah.
I believe it was over here. Iron deficiency, and it's thought to be a way in the old days that people try to replenish their iron.
So iron deficiency.
Is an association between those?
Because there's a lot of iron in many soils. Yeah. Oh, let me take the question right behind you.
Okay. Just on that one, poorly washed carrots help.
Poorly washed carrots. It's true. Good point.
My question, if on the risk of human manufactured chemicals to the world of microorganisms becoming maybe a really bad problem of some kind, how are the studies laid out now to try and assess or predict this? And if it turns out it's pretty bad, then what?
Okay, so the question, I think, correct me if I've got this wrong, the question is, okay, so we have all these human-generated or anthropogenic chemicals that are being released into the environment. How do we figure out whether microbes might be a potential solution to the chemical, or whether there's an impact, a direct impact on the microbial community? And if there is an impact, then what's the solution to that? So the figuring out the impact is not as trivial as one would think, because we can't bring it back. We can't like just go and be like, oh my gosh, acid mine drainage, I'll use that. Okay, so I work with a lot of acid mine drainage. I can't go out to a site and say the organisms are there or not there by just looking at it. So I have to bring samples back to the lab. I have to do some analyses to figure out which organisms are there, and then I'm either predicting their capability or their function based off of identity, or there's assays that I can do in the lab to see if they're capable of doing activity X. But what happens in the lab and what I see in the lab does not always translate into the environment. Because if the environment, as was previously brought up, the environment might be low in oxygen. In the lab, I provided a lot of oxygen because I'm trying to grow the organism. But maybe the organism isn't doing what we're hoping it's doing in the environment because there's something it's missing. Could be too little iron, too little oxygen, nitrogen, sugars, whatever it is the organism needs. So how do you fix that? Well, one of the things is that if you think there's potential from laboratory studies, you can fix it by modifying the environment, which we have some problems with. People don't get happy when we start putting in a lot of infrastructure into the environment. So to figure out the negative impact, though, is that still same relationship. So what we do is we have to look at the environment. We still will come back and ask, is there anyone there that's doing what we're hoping will get done? Then we go back and look at the environment, and then what we have to try to assess is, have we experienced loss? And that's hard because if we're not looking at a before picture, then we're just seeing the after, and it's very difficult to determine loss. Now, sometimes we can catch it in the progress of loss, in the process of loss. And then that, for sure, we can say, okay, there's a toxic effect, and then we have to figure out what that toxic effect actually is. But sometimes it's just guesswork on whether or not we even find organisms capable of, And if we don't, we assume that there's been a toxic impact. And if we do, but they're not surviving, we also look at potential toxicity as well. It's a really complicated and difficult situation to assess, for sure. Did I get there? Is that okay?
And the other part, because if we're about to...
Oh, what are we going to do to fix it?
To hurt the nitrogen cycle,
Well, so if we have so much chemical release that, let's say we actually confirm for sure, without a doubt, nobody's going to argue with us, that we're losing a critical aspect of the nitrogen cycle. What's the solution? How do we fix it? Is there a reverse? I don't know. I can't say. But I would just assume not get to that point. Right? So my solution is, let's fix what we can fix as far as like detoxification within the environment so that these chemicals don't persist, and then try to reduce the chemical load on the front end. Right? Because, you know, I don't want to get to the point where it's like, oh, crap. And we don't know what the solution is, so it's done. We don't even need to have a discussion anymore. Right? So I don't know what the fix is. Yeah.
Just on the mundane day-to-day, I eat a fair amount of fruits and veggies. I may want them under the sink, but how much washing should one do with fresh fruits and veggies?
Right. Yeah. You know, when I worked with Campbell Soups, this became very apparent to me why we have the foodborne illnesses that we do. And again, it's not at any fault to Campbell Soups. And the question was, you know, how much washing of fruits and vegetables is enough? You know, can you overwash, can you underwash, et cetera. So one of the things that happens with our food production, and this applies to fresh fruits and vegetables as well, is this commercialization aspect of it. We have so much product, and even Campbell Soups or Yoplait yogurt, et cetera. So whether you're talking a processed product or a natural product, the processing of it is so extensive and is so common. So all fruits and vegetables, once they get picked out of the field, for example, go into these giant vats, and it depends on the company, but of washing solution. Well, you dump enough fruits and vegetables in there, the washing solution itself starts to become loaded with microbes, to the point that you're not really washing anymore. You're now starting to inoculate. And all it takes is for, you know, a small batch of a contaminated food product, and because you have all the shared equipment, shared washing, shared packaging, the equipment itself can become contaminated, and it spreads. When I worked for Campbell Soups, I was really, really worried about Clostridium Botulinum. That was my #1 organism, because I was working on their tomato processing line. That was my #1 organism that I was watching for. Huh? Yes. Yeah, well, but it used to get into the tomato juice, and
That's the one that actually had the scandal.
that has the scandal. Yeah, the more public scandal. Yeah, absolutely. Absolutely. So Clostridium botulinum is common in soil. It gets on tomatoes. The tomatoes come in from the field. We have these really common washing vats. And then the tomatoes are moving through on conveyor belts. And they go to their different destinations. Tomato soup, V8 tomato juice, spaghetti sauce, blah, 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 blah. So whether they're going to be stewed and, you know, whatever. So all it would take is a small batch of tomatoes to come in, and now a lot of the equipment was contaminated.
But just the fresh ones on the counter to pick it up.
And so the same thing applies with the fresh ones, because they're also used on common harvesting equipment and washing equipment. It's really easy to have massive contamination. So how much washing do you have to do? I can't answer that. I don't know. I use a little bit of dish soap and water. And I use the dish soap to actually break down the waxy coating that's put on a lot of the food products. But I use water, and I physically try to disrupt the surface of the fruit or the vegetable. But I don't use any of the chemical washative additives that you see in the grocery store, because that's counterproductive for what I do. I don't do an overly aggressive washing. So I don't think there really is a magic number. I mean, I think they put out like a minute or something under running water is what you're supposed to use. And what is attached to the surface of a cantaloupe versus what is attached to the surface of an apple and a carrot is all going to vary. And the texture of the food product will also. Like I used to not ever wash cantaloupe. And then we had the outbreak from our plant up the street from us, right? And so now I actually wash it, and that one I do use a little more soap and try to aggressively get to the skin of the cantaloupe so that when I cut through it, I'm hopefully not just transferring. So a very inadequate answer to your question.
That's what you do, that's the main thing.
Yep. What else?
I have a question about,
there
you mentioned earlier the DNA of these microbes. And so I was just wondering, is it the same as with us and animals and plants or whatever, where we're both humans, we're the same species, but we have different DNA? Or with microbes, is it like this is the DNA of this type of microbe?
You just hit on a subject that we argue about a lot within the field of microbiology. So, okay, so I'm going to take a stab at it. All of us in this room, we all belong to the same genus and the same species. We're Homo sapiens. So the majority of our DNA is very, very similar. The differences genetically amongst us are very small, very limited, okay? If you were to look at bacteria and you looked at all E. coli, so there's your genus, Escherichia, and coli is your species. You would also find that the majority of the genes that you would isolate from E. coli, no matter where the E. coli came from, are going to be similar. We use one gene in particular to identify bacteria. It's called the 16S ribosomal RNA gene, which has a whole other suite of issues with it because you're relying on one gene to identify a species. That gene will be common amongst all E. coli. So now if you're looking at E. coli versus Salmonella typhimurium, they're not going to have the same identical genes. So we would be able to genetically distinguish them from each other. Where it gets complicated is this. Prokaryotes, the bacteria in particular, are very prolific at exchanging genetic material with each other. As a matter of fact, a study just came out that actually shows that microbes in different parts of our bodies are exchanging genes at levels that we didn't know. And there's actually genetic transfer that occurs across human blood. So my oral flora might be releasing genes that get picked up, let's say because I have a cut in the gum, gets picked up in my blood supply, gets traveled down, there's a cut in my GI tract, some of those genes come out with the blood in the GI tract, and some of those microbes pick it up. It's one of the mechanisms of genetic exchange. So that's the key. They're always picking up genes from their environment, and they don't necessarily know what the genes are, what they code for, and even who they came from. There's a lot of evidence that microbes pick up human genes all the time. There's also evidence that human cells pick up microbial genes. So that's just a biological phenomenon. But how many genes do you have to pick up before you start looking different than what your genus and species would otherwise characterize you as? And that we're arguing about because we don't have a good answer for that. As long as the core genome is still there, we'll still identify an organism as E. coli. Even if it's picked up, let's say, 1,000, which would be excessive, but 1,000 genes from some other organisms, it would still be identified as E. coli because the core is still there. But there is some evidence that when you pick up new genes and you start to behave differently because of those new genes, your identity perhaps should change. And that's kind of where we're discussing.
This is just a quick question. If you personally would ever take antibiotics for any conditions at all, or are they pretty much off your list?
Oh no, they're not off my list at all. They serve a very critical purpose, obviously. I am also, though, for me personally, and everybody has to make their own decision about this, I will do my darndest to not go in and see a healthcare provider. No offense to any healthcare providers in the room, right? But that's my personal decision. And if I do go in, now I usually have a pretty good idea if I have an infection, if it's bacterial or viral, but I have access to tools and stuff that other people don't. So I have gone in before, and I'll quiz my healthcare provider. I'm like, really, are you sure this is bacterial? Why do you think? What evidence do you have? Right, before I'm going to take an antimicrobial. But that said, I mean, these chemicals save lives, and they're really important. So there has to be sort of this personal balance, or maybe we could argue there has to be a societal balance between when they get used and when they don't get used. And those decisions are not as black and white as like what a lot of my students like to think. I'll never take an antibiotic. Don't say that. And how do you know when an illness is at the point where it really necessitates that level of intervention versus maybe your body still has a chance, your immune system still has a chance of controlling it? That's hard for anyone to predict, healthcare provider, non-healthcare provider. Yeah.
Yeah. What about prebiotics? You hear about probiotics and prebiotics. Do they have any value?
I'm going to be perfectly honest with you. I don't understand what the clear distinction is between a prebiotic and a probiotic. I think it's a lot of sort of commercial mumbo jumbo, in my personal opinion. The idea, I think, is this. You would take the prebiotic before illness, so I would take it as a healthy individual, which, by the way, many people do already with probiotics, which is where it gets confusing. And whereas probiotics, a lot of times people are talking about them as a potential outcome, if you will, of illness. So if you had like diarrhea, you take a probiotic to try to replace the flora that might have otherwise have been lost. That's sort of like the general public perception of it. So the idea is that the prebiotic would be pre-treatment. The reality is this: many people take probiotics regardless. Healthy, not healthy, illness, no illness, et cetera, et cetera. So my stand on the prebiotics is the same as my stand is on the probiotics. And I'm not sure that there's really a scientific difference between the two. Yeah.
So if I take my salad and I put it in a blender and I drink it, which is what I do.
Yes.
Before a bike ride, is there any degradation to not chewing it other than saliva and all the other stuff? But am I missing roughage? Because it's really good for a bike ride.
I mean, yes and no, right? I mean, even really finely ground up like lettuce, for example, is still going to provide some roughage. Are you going to get as much as you're actually going to have more access to the nutrients that are associated with ground up lettuce than if you were to take it in as bigger chunks. Because our bodies are, as humans, the human part of our bodies, we don't degrade cellulose. Our gut flora can, by the way. But they typically don't complete the degradation process in the retention time in the human GI tract. So that's why it can still provide roughage.
And then they degrade cellulose and a lot of B vitamins get created.
Yeah, so here's another fun fact. Humans, when we looked at the human genome, we're incredibly deficient in terms of the genetic components needed to make our own vitamins. Human cells, us as the human part, don't have the ability to make very many vitamins. We used to think it was all about diet that provided our vitamins. Now we know there's the microbial interface, because the majority of microorganisms can make the full complement of vitamins. So it's actually our gut flora that's making the majority of the vitamins that we need that then get absorbed by the body. So what you're doing by grinding up the lettuce, so to speak, is making more of those nutrients available for your gut microbiome that then in turn benefits you as the human part, because more of their nutrients and the nutrients exactly are being made available to your body. Okay? I'm happy to keep going. It's up to the group.
Probably the last question, because we're almost to 7.
Just getting back to the fecal thing. What percentage of fecal material is actually microbes, and what is food waste?
Current estimates, and it depends a bit on the individual and your diet, is anywhere from 60 to 80% of an average bowel movement is microbial biomass. And if you, just looking at the bacteria, if each bacterium has an average diameter of 1 to 2 micrometers, you can't even see it with the eye, that is a whole lot. of microbial biomass that gets wasted out in a bowel movement.
Would you classify that as a shitload?
Yes, thank you, yes. Using scientific terminology, that is a shitload, yes.
Yes, I think
Okay.
we should
Thanks,
give her a
you
big
guys.
hand.
Thank you.
Thank you, Timberley.